Warm welcome to the Professor R. Nagaraju sir, Padmavati University, Tirupati. And today, FDP program sponsored by AICT and JNT University, Anantapuram. And Ms. Netravani, the faculty member, JNT OTPRI, will introduce the today eminent speaker and very good intellectual, Professor R. Nagaraju sir. Netravani.
Yes, sir. Good afternoon, everyone. I am G. Netramani, faculty in Department of Pharmaceutics. Feel great pleasure to extend a charmful welcome to the eminent professor in the field of pharmacy, Professor R. Nagaraju sir, for this faculty development program sponsored by Faculty Development Cell, AICTE, New Delhi, on Pharmacy Education and Research, Present and Future Challenges, organized by Jntua OTPRI, Anantapur, and Directorate of Faculty Development and IQAC, Jntua, Anantapuramu. Professor R. Nagaraju sir is currently officiating as Professor Dean, School of Sciences, Sri Padmavati Mahila Vishwavidyalayam, Women's University, Tirupati, Andhra Pradesh, and having 20 years of research experience and 33 years of teaching experience. He obtained B.Pharm, M.Pharm and PhD from College of Pharmaceutical Sciences, Manipal. Dr. R. Nagaraju sir started his career as a lecturer in Vishweshwarapura Institute of Pharmaceutical Sciences, Bangalore in the year 1986. Dr. Nagaraju sir joined as professor in the Institute of Pharmaceutical Technology, Sri Padmavati Mahila Vishwavidyalayam, Women's University, Tirupati in the year 2004 till now. Sir obtained grants about 237 lakh rupees from DST, UGC and AICTE. Professor Nagaraju sir is awarded with 2 patents in dental implants. He organized UGC-sponsored 3-week refresher course and national seminars. Sir has guided 43 students for M.Pharm and 10 PhD students, and 8 students are presently working for PhD under his guidance. Dr. R. Nagaraju sir is R&D consultant to various organizations and member of Governing Council of few educational institutions. Professor Nagaraju sir published about 73 research papers in reputed national and international journals. Sir delivered about 126 invited talks in various national and international conferences. He is an editorial board for few reputed journals. Sir held many administrative posts in Sri Padmavati Mahila Vishwavidyalayam, such as Dean, Development Head, Director, Center for University Ranking, Dynamic Director, Directorate of Distance Education, Chairperson, IAAC, etc. He received, regarding his awards, he received Best Pharmacy Teacher Award by Association of Pharmaceutical Teachers of India for the year 2018. He is the recipient of State Best Teacher Award 2017 for the subject Pharmacy by Government of Andhra Pradesh. He received National Best Teacher Award 2017 by CVS Krishnamurti Charities, Tirupati. Sir also received Best Teaching Pharmacist Award 2017 by Indian Pharmaceutical Association, Tirupati Local Branch. Sir received National Level Pharmacist Award 2017 in the category of Scientific and Innovative Research Award 2017 by Simandra Drug Dealers Association and Andhra Pradesh State Pharmacy Colleges Management Association. He received Pharmaceutical Scientist of the Year Award 2017 by Andhra Pradesh Council for Science and Technology, Government of Andhra Pradesh. He is also the recipient of Distinguished Alumna Award of Manipal College of Pharmaceutical Sciences, Manipal, for the year 2005. He is also the recipient of the Achiever of the Year 2002 Award by Avopa, Bangalore. We are very happy and eager to have your lecture, sir. Thank you, sir.
Thank you, Ms. Netravani, for your kind words.
Thank you, sir.
Professor Gopi, can I start share the screen?
Yes, sir. Yes, sir. Proceed.
Yeah. Bear with me for a few minutes. Are you able to see
Yes, sir.
the screen now?
Yes, yes.
Yes. Yeah. At the outset, if any time in between there is some disturbance, please let me know so that I will try to correct it. Right. At the outset, I'm thankful to Principal Dr. Gopinath for his kind invitation to be a part of this particular program. And also, I'm thankful to all the, I can see in the participants, few of my old students and also few known faces. And it is very much essential to, you have taken up your time and participating in this program. The topic given to me, I've been asked to speak on the novel drug delivery research. So this was actually up for the topic what already Professor Gopinath has got sanctioned from the UGC, I believe the ACT project, no?
Yes, ASTD.
ACT, yeah. After the Sriram's glorifying lecture and energetic one, probably, I'm taking to you, he has purely on education he has given, on the problem-based learning and how it is going to be important and how the recent advances. So with the education now, let us move on to the research. That's what your theme says. So I selected the novel drug delivery research because I am from technology side. So I thought I can introduce or I can give a little of my experience in this particular area. I gave the title as Emerging Trends in Novel Drug Research. So it's very important. I would like to start with this word. The secret of good sermon is to have a good beginning and good ending, and the two should have too close as possible. So I believe in that. I don't take much of your energy to see. I wanted to open and close it as soon as possible within 45 minutes or so. So all of you know that I am proud to be associated with Sri Padmavati Women's University. It is popularly called as Padmavati Mahila Vishwavidyalayam, a government university, the only university in the state of Andhra Pradesh and Telangana for women, and one of the universities in South India, the first university in South India. I am in this university in the Institute of Pharmaceutical Technology as a professor of pharmacy. And I'm proud to say that our university got 150 to 100 to 150 rank in the as a whole university is concerned, whereas NRF ranking for pharmacy is quite impressive. We have got last year 47, and this year we have got 42. We have improved upon it this year. The current year we are expecting we are going to improve upon this. This 42 number, it is an Indian ranking, all India level, and I'm very proud to say that we are the second in the state of Andhra Pradesh. Of course, the first is going to be always, I should say, Andhra University. So probably to reach Andhra University level, I don't know when we are going to beat, but anyway, we are trying our level best to reach that. I'm very proud to be associated with such an organization, and where we have got plenty of opportunities to grow, and also our students. Right, as soon as if those people who have entered the university, you know how it is the university. This is an old picture. Why I kept this picture is these flowers, now it is not there. So that's why I kept this picture. Now the building is very nicely done. Good facelift was done, and good thing is next time I will take the current picture also. But this was the picture when I joined in 2004. It was there. So for the sweet memory, I kept this one only. It used to be very nice in the campus. Those people who are visited the campus will definitely agree with me. A sprawling 121 acres, 123 acres of land with greenery, with the lockdown period, we used to see so many birds in the evening, 5 o'clock, 6 o'clock, we go around the campus. We see so many birds because of the no students and pollution-free environment, and all that nature is favoring. Probably the nature lovers will have a very good insight in the campus. The university was established in the Act of University Act 1983, a statewide university. This is exclusively for Women's University, accredited by NAC. Now we have got A. We are trying for A+. The theme is emancipation of women through acquisition of knowledge and empowerment through skill upgradation, involvement, and participation in various occupations of the society. This is the brainchild of the erstwhile CM, Nandamuri Tarak Ramarao Gari Manasa Putrika Antaru University. So he has done a great way that establishing a student university for women, the two in a backward area in Rayalaseema, the two in Tirupati. And we have on campus now around 6,000 students who are studying various courses, and the 5-star courses are, needless to say, pharmacy. And then recently we started engineering already 5-6 years back. Now it's also very popular. Engineering college is very popular, and we have nursing college a few years back, and we are starting MSc nursing also. And we have all other courses, courses in the science, starting from biotechnology, microbiology, sericulture, in the arts, Telugu, English, and women studies and social science. There are so many wide variety of these courses are available for the students to pursue their career and come out emancipation of the woman. That is very important. And in the current scenario, you see now the woman empowerment is very important. This was realized 33 years back. That's why Chief Minister and given this university. I am very happy to be part of this university. And after 2004, all my students are women only, young girls in B Farm and M Farm, and women in PhD, and they have been shown a good progress in their life and has been doing very good in their profession. Coming to my presentation outline, even though to the gamut of this particular audience, I need not go to the very basics of it. But anyway, the set induction is very important. So I'll try to give some brief introduction and of course advantages of this novel drug delivery research. And first of all, why this novel drug research is being focused. And then the current recent trends, there is a nanoparticulate drug delivery systems. And I have taken few slides I will spend on liposomes, a case study. And of course, coming to the challenges, what as a young scientist we are going to face and you are going to face. And in a bird's eye view on, aeroplane view on, novel drug delivery research in our own university, and finally concluding with my remarks. So all of you know very well, as all of you are teachers, I did not give much of the basic information. It will become too much, or say it will be you will read 3:30, you will try to sleep. So I just touch upon only the very fundamental aspects in 1 or 2 minutes and then go to my topic. The novel drug delivery systems, novel means new, not conventional. These novel drug delivery systems has been looked upon in the pharmaceutical research in recent times, mainly to see that the potency has to be improved, drug potency, and to have a controlled drug release, and also to obtain sustained therapeutic effect, and to provide greater safety than that of the normal dosage form. And finally, if it is possible, to target the specific, the drug specifically to the desired tissue. So with these sole objectives, the novel drug delivery systems have been conceived, and which is not conventional, they are considered as novel for any part of it, whatsoever it is going to be. So these novel drug delivery systems mainly focuses on evolving various approaches and developing various formulations and technologies and systems for transporting the drug in the body and where it is required to achieve its desired therapeutic effect. So that is going to be very important. What can be specific advantages of this novel drug delivery systems over conventional formulations? What is wrong with our tablet and capsule? What is wrong with our normal injections and so? There is nothing wrong. It is only an improvement, and what we are going to look into. Enhancement of the solubility and bioavailability. All of you know very well that most of the drugs, what we have in the developing or developed drugs, are having the solubility problems. That is, it become, most of them belongs to BCS class 2 drugs. So having a poor solubility and good penetrability. So if we just try to address this problem, we will definitely improve upon the bioavailability. And we also see that by preparing novel drugs, novel drug delivery systems, we will even attempt to reduce the toxicity or protection from the toxicity. This problem is a very important burning problem in the conventional drugs, and where we can address to some extent by preparing the novel drug delivery systems. And by doing so, sometimes we can even think of enhancing the pharmacological activity. Therefore, if enhancing the pharmacological activity leads to decrease in the dose. So when the dose is decreased, decrease in the toxicity. When the dose is decreased, further cost is going to be effective, and so on. Major problem is we can even design a dosage form which is having a better solubility than the conventional dosage forms. So with these specific objectives in the mind, we do work in the novel drug delivery area to bring out for some new formulations what is going to be. And the current research in drug delivery systems goes mainly on altering the route of drug delivery, thinking of various alternate routes of drug delivery, using different types of delivery vehicles, what is going to be possible, and using various targeting strategies, active targeting, passive targeting, first order targeting, second order targeting, and so on. And the medications can be prepared which can be taken by swallowing, normal one, by inhalation, by absorption through the skin, topical preparations, or by intravenous injections. So for novel drug delivery drug delivery systems, there is no limitations in any way. We can think of novelty or developing new drug deliveries in whatsoever, whichever where you are going to go. So why in the current scenario, the novel drug delivery systems has attracting a lot of attention? All of you know very well, or majority of you are teachers. Some of them are doctorates also and experienced also. You know very well, when you see the development of a new drug molecule is very expensive. It is going to cost more than $120 million, and it is a time-consuming process. It is not just it is going to come. It is going to take a decade or more. Even though you spend a decade or more time, there is no guarantee that we are going to come out with a drug. So my professor always used to tell the research or drug development is one where searching for a black cat in a dark room. I also tell my students about the research. Research is something where you're searching for a black cat in a dark room, especially when the cat is not there. So at the end of the day, we are going to, after 15 years of spending, we may even come out with fully negative results. So that is the risk of developing a new drug molecule. So in spite of the risks, only the companies are going to go. India is the one where we will not be able to pursue the drug discovery. We're not doing good in that area because a lot of money is required, and we don't have so much of funding in development of the new drug. That's why we are very good in novel drug delivery systems, and also the generic drugs, which where our Prime Minister is promoting, Prime Minister even promoting the generic drug development. Even countries like America, they are also wanted the generic drugs, which are made in India for America. So the novel drug delivery systems also have an attention, also have been attracted the attention for safety, improving the safety and efficacy for the old drugs. That is very important. We are using the old drugs for the new applications, the old drugs for the better characteristic features, better activity. So therefore, we can also think of the individualizing the drug therapy, and we can go for dose titration and also the therapeutic drug monitoring can be easily done by doing this. And we also think of attractive methods where the drug delivery, delivering the drug at a controlled rate where it is not possible in the normal conventional dosage forms, where we can also deliver the drug at a very slow rate, where sustained action and targeted delivery. So modify the release profiles of the drug to suit the requirement. And again, is the drugs which are already there in the market. So drugs which are having the proven efficacy, proven safety, and so on. So this is where the new drug delivery systems has conceived the ideas how it is going to be. Further, to add to this, when you see closely, we see the very slow decline, or I should say, a drastic decline in introduction of the new chemical entities since late 1950s. So we are whatsoever drugs we are getting, or the blockbuster drugs, what we talk about, majority of them are invented before 1950s, and afterwards we have few drugs, but not in the same pace what in the previous 1950s. And also we see, because of a lot of money involved, only few firms are conducting this type of drug discovery research. So that is also because of that, with the new drugs are coming into the world market very low. And we also see a positive way erosion of the effective patent life. With the drugs patent life is come to the generic market, and the generic market drugs are much more cheaper and therefore available for the people. You can think of taking novel drug delivery systems using such drugs. And also, when you see the US market, always we compare the US market, novel drug delivery systems may account for as much as 40 to 50% of the US marketed products. So compared to India, the acceptability and the approvals or the drugs, novel drug delivery systems are almost 40 to 50%, and whereas that particular percentage is comparatively less in the Indian market. Because of the reason one I see, because the economy, the cost of these novel drug delivery systems are very high, and we're not able to purchase, the common man are not able to purchase and afford the cost. So whereas US market, everything is very well insurance driven, so they don't mind how much money they are going to get. They are going to reimburse from the insurance company. I was just now previously talking about controlled release technologies. Controlled release technologies not only effectively used in the development of the oral drug delivery systems, but also it is used in the development of the transdermal and transmucosal system. There is a lot of research going on in this area, and there are few products in America in this particular area. We also see the nasal and buccal aerosol sprays are also taken a lot of attracted a lot of young researchers to deliver the drugs through the nasal route and buccal route. And the drug impregnated lozenges are also is the day of it, and there are 1 or 2 products are there in the Indian market also. So therefore, the lot of growing interest in the people are stepping into the novel drug research, not only in the academic one, but also in the field of pharmaceutical industry. They are working hard in bringing those drugs. But compared to generic drug development, the novel drug delivery research in India is a bit slow. That is my findings, where we see this particular area is comparatively slowly things are happening. And the controlled release technologies, the new versions are going to be the encapsulated cells and oral soft gels and ionophoric devices, where it can be used administered through the skin without piercing. And we can also use programmable devices, where programmable insulin devices are available, and where programmable and implanted, the drugs can be subcutaneously with a minor surgery, they will implant and try to do it. This is also a lot of work is going on, and few products are there in the US market, where they focus upon developing the novel drug delivery systems by the controlled release technologies. So these are all various aspects of it. As I was telling you, to this audience, I need not talk about the fundamental aspects such as how do you prepare the liposomes, how do you prepare nanoparticles, how do you prepare nano devices. So all those things you are very well teaching and you are very well in the research. So what I try to focus upon is going to be what is the current trend and what is the possible focus which is required. So the future prospects of Ndds is very broad area covering which is areas related to nano devices, nano medicines, nanoparticles, nanosomes, transferosomes, neosomes, and so on. So basically the nanotechnology has been commonly used for the development of the drug delivery systems. People say the nanotechnology is going to be a very promising field evolving over a period of time. This is actually a slide which has been done in 2001. You can imagine now we are in 2021. Even after 21 years, this particular information is very valuable. And we see the growth innovations, textile innovations in 1971 to 1953, 1853, and rail and road development. This is not in India. This is in majorly the developed countries. So please don't sink your understanding with Indian situation. Railroad developments, before the first line has been railed in India, there was already super fast trains available elsewhere in the world. 1825, it lasted up to 1913. Then automobile, various cars and other things have come, and it has gone. The peak was gone 1969 and still is going on. We see it is lasting still. And then you see the most very important innovation what we have seen in our life is a computer. Yeah, computer we are dealing here. This person, Milonovich, person told in 2001, the computer is going to be ruling the country, ruling the world beyond 2025. Definitely, it is true. Predictions are true. We are now in computer innovation, growth innovation. Now in the computer, we have seen now artificial intelligence, we have data management, we have big data, whatnot, everything. We also see robotic technology. So many things, advances are we have seen now, and we are seeing using the artificial intelligence in drug development, artificial intelligence in various parts of the pharmaceutical things are coming up with the researchers. And of late in what you call 2000, it is started the nanotechnology, and they predicted it is going to last the whole what you call the whole this 2000 up to 81 or beyond that. So now we are in this era of the growth innovation of computers and nanotechnology. I am not limiting the nanotechnology to only pharmaceutical. Nanotechnology is a technology which can be used in different parts of the what you call developments, but I will limit my discussion only to the pharmaceutical technology, how it is going to be useful. So the nanotechnology has been very commonly used, and which has been entered into the pharmaceutical research in making or designing of the drug delivery systems. And again, this slide is very basic, only to impress upon you, to bring back your memories to in nanotechnology. It is nanotechnology derived from a Greek word, nano meaning is dwarf, and this applies the principles of engineering, electronics, physical properties, and material science and manufacturing at a molecular level and submicron level. So the particles, when it is being reduced the size range between 0.1 to 100 nanometers, it is exhibiting, it is behaving differently than that of the larger size. So that properties are going to be explored here, and that is going to be studied, and that is used in the development of the drug delivery systems. But however, if we by the book it is say that for the all practical purposes we say 0.1 to 100 nanometers. In literature we can also think about up to 1000 nanometers for the pharmaceutical nanoparticles. The only thing is here, we all recollect or say understand that the particles when you are there in nano range show completely different physical and chemical properties are same. It is going to have a different physical properties that is exploited in this particular way. So the nanoparticles are attracted so much cutting across various fields of pharmacy and also other fields because of the methods of preparation are found to be very simple. And this I have found some colleges, even I visited some colleges as an examiner, as in MPharm or PhD thesis. They are doing, in one of the colleges, they are doing the nanoparticles at the final year level. So nice, you know, the final year students are preparing the nanoparticles in the lab. So it is very easy. All of you agree with me. It is very easy. This is called as a stretching beyond the 4 brackets of the 2 brackets of the syllabus. So this is what where anybody look into innovation in that particular college. So the students in the B form passing out that particular college will definitely have a better understanding and attitude than where there nobody is doing any lab and only writing the record and giving the marks. So what I mean to say here is the point here, what I want to stress upon is very simple, even you can do it in the B form laboratory. So that's what my point here it is. So we can do it, and very easy to scale up also. Scaling up is one of the very important tasks. Once they may be successful in the laboratory, they may be successful in the pilot plant, but scaling up in the pharmaceutical industry probably really altogether a different task has to be managed and challenges are there. So therefore, these nanoparticles have got that particular advantages where we can go ahead. That's why many of the researchers are going ahead in venturing into the preparation of the nanoparticles and exploring the properties of the drugs and the dosage forms in a way what we are going to look into. And these nanoparticles are found to be very stable, and if you formulate properly, and also easily freeze dried. So easily freeze dried, my point here is they can be converted to the powder and the stability is further increased. So handling a powder is very easier compared to the other things. So this is also found to have a successful tissue targeting. So the tissue targeting, the point which is where the conventional dosage forms, we are not able to achieve this particular tissue targeting. Where we can achieve this tissue targeting, we can make antibiotics nanoparticles. We can go for preparation of the cytostatic static drugs and nanoparticles, and the larger molecules, the peptide and proteins and nucleus. We can target these drugs into a place where you want in the cell targeting or inside the cell targeting possibility is there. So that is the versatility of the nanoparticles, what is going to be. And also these nanoparticles have the excellent stability, their protection of the drugs against the chemical and enzymatic degradation. So because of this, the enzymatic protection against enzymatic degradation, it will not metabolize in the during the course of time it is spending in the traversing from the site of the absorption to the targeting site. So therefore, enzymatic degradation is not there, or is going to be, that is going to be, the products is going to be stable. And for majority of the drugs, the nanoparticles found to have reduced side effects compared to that of the particles which are in the normal size range. So this attracted the people to go into the nanoparticulate drug delivery system, venturing into the drug delivery system, the nanoparticulate systems. So I have tried to list out some of the nanoparticulate nanotechnology based drug delivery systems where liposomes, I mean, don't get panic that I am going to take a class like a B.Pharm or M.Pharm students. I'll only touch upon the very important points here and leave it to you. You are already teachers and come a long way and spending a lot of time in the research and other things. So the liposomes, and for a thing, Dr. Gopi suggested me to take one small case study. So that's why I'm taking a case study of the liposomes, and remaining thing I may leave it to the audience to go for the further thing. I may also touch upon a few important things what currently in IPT, what we are doing in nanotechnology research. Liposomes, neosomes, microemulsions, nanoparticles, nanosuspensions, solid lipid nanoparticles nowadays attracting a lot of research. Nanogels or hydrogels, microspheres, micellase, right? So liquid, it is also called as cubosomes. So where the micellase are going to be used and then nanocrystallizations. So likewise, there has been a lot of what you call research going on in this particular area, and each one of them has got its own advantages and disadvantages. And as I just now at the beginning I mentioned, I'm not going to go the technical issues of it, how it is prepared, what is the methods of preparation, what is the methods of evaluation. All will be boring to the audience like this. I'll only touch upon how important these are going to be. So I'll take one example as the liposomes. Many a times when I teach the liposome, I started teaching liposomes way back in, started in Rajiv Gandhi University. They introduced the liposome novel drug delivery systems in final age itself, way back in 2002 or 3 or all. So that time, liposome were a hot topic. Worldwide, a lot of research was going on. And over a period of time, over a period of 15 years now, we see a lot of things happen in the liposomes. And I used to say previously, a liposome is something where very good at laboratory cannot be successful in the industry, or cannot come into the market so easily. But all that has been come out now. Liposomes have been successfully in the market, and the liposomal preparations currently are there. And I think I don't require to tell you the liposomes, lipid particles, vesicles, and all that. All of you know about it. So you can see here the various drugs has been tried for the liposomal preparations and various categories. What is being shown here and what has been circled here has been already entered into the what do you call clinical studies. There are currently the clinical trials, and some of them, probably this slide was I have taken from the one particular reference here. This was 2 years back. I did not get current information on that. Sorry for that. So maybe 1 or 2 years back, this was the information. So the liposomes were successfully prepared for the wide variety of the drugs in the following categories, like anti-cancer drugs, antibacterial drugs, antiviral, DNA material, enzymes, radionuclide, and fungicides, and vaccines. So when you see these compounds which are at the star around, doxorubicin has been on the clinical trials and current clinical trials, and may hit the market for commercial use at any time. Likewise, you see the cDNA and CFTR, and this is the DNA material which has been commonly tested for this what do you call the efficacy in the form of the liposomes. And you can see here antibacterial drugs, clindamycin, ampicillin, the very traditional drugs, rifampicin has been tried to do. They are developing liposomes, and already they are under the way. You have got a lot of these are the things where the industry is working on liposomal drug preparation. I am not talking about the educational organizations or academic institutions where we are working on. This is what the liposomal drug preparations is strongly pursuing by the industry, and where at any point of time, some of them may enter into the clinical use. And the fungicides, amphotericin B. So this is the status of liposomal drug preparations. All of you know liposomes are prepared very easily in ether injection method, solvent evaporation method, and we have got different types of liposomes: single layer liposomes, multilaminar liposomes, unilaminar liposomes, and so on. We are going to evaluate for particle size. We are going to evaluate for decay studies, drug content. All that is all there. Those are all the science behind that, and the technology in the industry has to be developed. That is a major challenge. And I was just now mentioning to you, the making laboratory success may not be successful in the pilot plant, and further may not be successful. Even though it is successful in the pilot plant, in the large scale production, it is not going to be successful. It is the convergence of the technology and as well as the science has to be converged to make it a product successful in the market. This is another slide where I got from literature. I have duly acknowledged. I'm very careful. Don't want to go for any plagiarism issues. So this is one of the way where if you quote the where you have got, so that means it is not mine. So this particular slide tell you about what are the medical applications of the liposomes which are there, and they are already present in the market, right? So developed by Milans and anti-substance and amend again 4 or 5 products from Milans, and cholesterol lowering, stimulate the appetite, topical estrogen therapy, anti-nauseating drug, and anti-immunosuppressant. So likewise, these are all the products which are underway and going to medical applications and were specifically developed for that particular purpose. So therefore, it is very important that we have got the plethora of nanoparticulate, nanotechnology based drug delivery systems where worldwide a lot of research is going on, and you know why this particular research is important, you know how this can be easily done, and so on. Similarly, for each and every nanotechnology based compound, since you have seen here, we have the literature backup and we have the products, and majority of the thing, apart from a little bit of micro emulsions and microspheres, I didn't see any references only in the literature, means only as a part of the what I should say, academic research. So industrially, people are not gone into that level. So that's why I'm not insisting, I'm not going to go in details of it, but this is the potential areas where we are going to go ahead. So with this, I come to the very important challenge what I'm always mentioning from the beginning. It is from the challenge from the laboratory to the pilot scale. So laboratory to the pilot scale should be successful, from pilot scale to the full scale. So this is the where you see a big challenge, and you may be successful in the laboratory. You may or may not be successful in the pilot scale. If you are not successful in the pilot scale, it will stop there itself. Like we have got the what is that? Particular systems where the RBCs are loaded, into the drug, and those are not confined to only the book. It is not coming to the day of life because it is very difficult. Likewise, we confine only to the laboratory. If by chance it is successful in the pilot plant pilot scale, successful in that case, then the full scale commercialization. So these are all the challenges associated. As an academicians and researchers, we can put all our efforts in development in the laboratory, but taking into a pilot scale require altogether a different technology and different expertise, where the product has to be taken and people has to work a lot and to make it success. And laboratory wonders ultimately predictated the research community's ability to bridge the gap. Some might say that between the science involvement and engineering required, particularly during the scale-up. So the big challenge, we may be successfully doing everything in the laboratory, but coming to the success in the market, success in large-scale production probably is going to be a big challenge to us, where you require engineering skills, and people has to bridge the gap between the invention, the science involvement and engineering required. This is a major challenge. Yeah, I remember that resealed erythrocytes. Resealed erythrocytes are popular in the laboratory wise and very good, and then it is not come into even to the minds of various industry people, because the blood is involved in that, and also the technology is complicated. We cannot go for a large scale. So likewise, many inventions are very good in the laboratory level, will not see the day of the light. Light of the day, I should say, over because of the limitations what it is going to be. So therefore, this is what the new education policy. I'm sorry to bringing the new education policy in between, because now maybe this is not that much very bothering about us, for me and all people like me, because in 3 years I am retiring, less than 3 years. So whereas the young people who are listening, the new education policy, you like or not, it is imposed on us. So where the academic industries, academic institutions, including in the future, we will not have any affiliating in universities. Each and every college should be it is going to go into the, what I should say, autonomous, and then NAC accredited or say NB accredited as applicable, and the degrees will be issued by yourself. So when the new educational policy insists on such type of development, and that's what this we are being, government universities has been forced to bridge the gap between the science what is available here with the engineering required. So we have been asked to go on various MOUs, transfer of technology from our I should say from the laboratory to the commercial scale. What is going to be the gap? So that's what our university is actively working out on the MOUs, what is possible, MOUs are possible nearby higher education institutions, and also the skill development component has to be there in the students. So where already PCI to some extent is doing it, but that is not sufficient as per the NEP. So therefore, the challenges in education are very challenging in the coming years, where probably we are shifting from a conventional to the more applied model, and where it is going to be very difficult for the youngsters like you who are going to transform from one model to the other model. Right. So the challenges are many, and the success in the laboratory does not mean that the success in the elsewhere, especially the commercialization. So a couple of minutes, I'll spend a few slides on giving our own institution, where actually we are working on this novel drug delivery systems. And these are all the people I took over the last 5 years. And this is a self-emulsifying drug delivery systems we have worked on. And Sarita, she got in 2015. And Umarani, still she's there in the audience. She successfully completed last year a gastroretentive drug delivery systems, and she did on micro balloons, a wonderful work which has been appreciated both by all the examiners, and she recently passed out and she got the degree. And she's working out to bring out at least one patent. I said to her, because that is going to be, she worked on gastroretentive micro balloon technology, and she had done excellent good work. And she's in the audience. Uma, thanks for coming to this to say a few words about you. Then we have developed with Pratyusha. She's also got last year 19, where the pulsatile release tablets. So this was another interesting area where I was looking into. A lot of challenges are there, and she took almost, both of them took almost a lot of time, 6 to 7 years I spent in developing this, but it was not a waste. Their work has been appreciated elsewhere, everywhere, and they got very good publications, and where I'll be receiving so much of citations for these publications. And then we see the colon drug delivery system development, where Priyanka, what is a full-time scholar, she got AGC scholarship, and we made here Nano Sponges as one of the colon drug delivery systems. So the Nano Sponges were tried here, and for one such drug, and it was successful highly, and that is also we are applied for patent for this, and waiting for applying and waiting all that 3 to 4 days it will take. And the interpenetrated hydrogels for controlled delivery has been a student naming Ramya Krishna is pursuing, and probably this year end or so she is trying to submit. And Jhansi Rani is basically we are given a broad title here, nanotechnology based drug delivery systems. She is trying 3 different nanotechnology based drug delivery systems, and therefore we will be able to come out with a good result. And then at the PG level, who are more active because we get more students. I get almost 3 to 4 students every year, and very dedicated and nice girls, and they won't go even home for weekends. They all wanted to work, best presentations. And Professor Gopinath has visited our college, seen how about our girls, how it is going to be. So these are some of the things, noteworthy things I have put here. And we have developed organogels, because nanogels and hydrogels are there. The organogels is one nanoparticulate based drug delivery system in an inform level. So metronidazole, this is also published. And I was talking just now about the cubosomes, the liquid crystalline nanoparticles. And this is done by Naveen. Now she is a full-time PhD student here in our own university, and she got the best presentation award in 2 of the conferences what she presented. And it is a cubosomes, liquid crystalline nanoparticles. And then we have prepared chitosol nanoparticles focusing on colon-specific delivery. This is Nagalakshmi, and her presentation was also appreciated, and she got the highest marks in that area in that time. And usually I don't take marks as a criteria, because the marks is not the criteria elsewhere in our life. Marks is only a one particular phase of it. The knowledge is important, the attitude is important, the success based on only that, not on the marks. And this girl has made nasal. I said no, that's why I made it. We made a micro emulsion in the intranasal applications. So this was again given we took the Professor Prasad help for pharmacological work for this, and these are all under the PG level. I want to bring it the notice of all the listeners here. This is all at the PG level. What works we have done under my guidance. We have 4 such pharmaceutical technology and pharmaceutics people. They are also working in different areas. And we also not stopped there. And nano suspension for ocular we have tried to use. That is a naproxen. And further, we have prepared a nanoparticulate scaffold that is for wound healing we have done. So this involves the wound healing. Animal work is involved in this, and where the scaffold are impregnated with the nanoparticles, what we have prepared the fluconazole, and we have got very good response. And this particular work also, actually, I wanted to continue for PhD, and this girl, I mean, due to various personal reasons, she didn't join. And then the next one was, what I liked was the nanocrystals prepared by Shruti, and where we have successfully prepared nanocrystals, and all the students have done the laboratory work in the SPMVB campus only. We are not sent them to any industries or any other places. Whatsoever facilities available, and of course we have got very good facilities available in our campus. We have designed the work, and successfully they could do. And then nano sponges, I was talking to you about. We have tried these nano sponges for wound healing, and again we took a natural drug for this, because I wanted to try. The curcumin has got very good properties, various properties, and this was again a success story. And then this girl, of course, this girl, she wanted to do in industry, and I sent to the industry, and she prepared delayed release tablets, and she could do, and she has also got this job in the same industry, and she's working there, continuing in the same project. Then we see, ML gel. So this is what I wanted to see here. You might have seen, I have not continued the similar type of work to subsequent students. I try with my M.Pharm students a different concept, and they pick up and they continue and come out with interesting results. And we see the psoriasis is a very, very disorder where the ML gel preparations, the first of its kind, the curcumin, again we have seen, we have made it a complex in the beta-cyclodextrin, and that one has been introduced in the ML gel, and we have got an in vitro model. We are not done in the rats or whatsoever it is. In vitro model is available that we validated and we have got it. And unfortunately, we could not, go for some of the studies where that's why the paper is still pending, and the next students will be going to do this. And this moxifloxacin nanoparticle, this has been published already. As soon as the first communication only, we have got it published. And this is the H2 gel. What we've done is moxifloxacin, we converted it into nanoparticles, and then these has been put into the gel. So it is a sol formation. Once in contact with the ocular, it is going to go for the gel. So it is a two concept: preparation of the gel and preparation of the nanoparticles, and merging these two for a better effect. So this paper got very good citation also, and almost for last 1 year, I see more than 100 citations is there for this, and she could do it here again. Further, I want to take it. Some other antibiotics are going to do this, and then we have done again floating in situ gel for oral, and for last year students I didn't get the data, and the time was also less. So That's why I could not include the last year students. In that last year, 2 students have done the nanoparticulate systems, and I didn't get the titles of them because of paucity of the time, correct titles. So this is what floating lanceable floating in situ gels we have been doing in this level. So it is going to be very important that using the technologies available and catching up the thrust areas and coming at least as to have a base for the going into the next stage. Next stage is now giving all the information in a nutshell for the people who are working in the industry. And if any industry wanted to take up any of the preliminary work, what we have done, we are going to give them the having an MOU, and the remaining part of the work we are going to do. That's what exactly the NEP wanted to do to us. So we are in search of the industries. We are working in these areas, not only stopping here. And as I always say, the sword is hanging on our head. We are expected to perform. There is no excuse. So we are in search of the people. Few industries are responded positively, and they have come forward to take up 2 or 3 projects, and they wanted to do the pilot studies. So therefore, it is going to be very important to us to catch up the field, and there is so much where the young people, the researchers can do, including you. You are being in a teaching profession. Many of you, I can also see Dr. Vijay Bhaskar. I know him for last so many years, last 20 years, I think, 20, 25 years. Welcome. So this is what the thing, and with a few 2 or 3 more slides, I'll try to come to the conclusion part. What the world needs is not like this, needs the quality. Whatsoever you do, the work should be quality, should have a quality. You should have a quality teaching, you should have a quality life, you should have a quality research. It is not just simply for somebody's take you need to go. If you have quality, definitely you are recognized, you have been promoted, you have been given all that things what is been required from the society. And the hard work is very important. The quality will not just overnight, it will not build up. You need to do the hard work. Hard work is sweeter returns, smarter returns, and the faster returns. Many times the young people who are sitting may be thinking, sir is no problem, he is in university, he can say anything. But we are in the Corona time, we got 50% salary, and we don't know whether the salary is going to be there in the future. All those things, all those problems are there. So there is a risks are associated with all type of things what you are going to do. By simply sitting, you're not only wasting your mental things, and you're becoming older, and probably when everything is favorable, you may not be active enough to pursue the work. So what I suggest is to you, even though the conditions are a little tough, I should say it is tough. I know a few colleges are terminating the, especially the PhD scholars, because they can't pay money. But that is a different story, whatsoever it is going to be. But what I tell the youngsters here who have a lot of future, you are going to be a part of NEP, national education, a very ambitious national education program, educational policy, where probably you need to come forward. In the days of when we were teachers, we had a different, altogether different days, challenges. Now you have a different challenges altogether. You need to come ways and means by which whatsoever we are good, you like, put a good hard work in that. Smarter returns, and of course the faster returns. And you should have 5 important things like credibility, commitment, quality, transparency, and the trust. So then only you are respected as an individual. If any of the things you are compromised, you will not, the society will not believe you. So therefore, try to have the balance of all this in such a way that you are going to come out you are a perfect individual. And therefore, I should say it has taken almost 30 years for me to bag so many prestigious awards. Till 2017 or 18, I was not, my application not enough, even enough. I don't have enough credentials to apply for any award. So after 30 years of work, what I have put up on recognition, especially when I came to, after coming to university, I have a different outlook, and where I have been working in a different thing, where I got the confidence that yes, I can apply for this, whatever it is going to be. So that was the thing. It is not just overnight. We have to work, and in the difficult times, you have to work in such a way that definitely the things are going to be better. Yeah, some mind a thing for you. Look at this picture, and what do you see? What are the images you see nearby? What are the images you see far away? Take a minute, 10 seconds, and anyone can unmute your mic with the permission of the organizers and tell me. Anyone? Audience? Audience, Chapani. 45 1 hour కూర్చున్నారు కదా ఇది లాస్ట్ ఇది అసైన్మెంట్ గోపినాథ్ గారు వీళ్ళకి ఉందా పర్మిషన్ ఉందా
ఎస్ సార్
వీళ్ళకి అన్మ్యూట్ చేసుకోవడానికి
పర్మిషన్ ఉంది సార్
మరి ఎవరన్నా చేయండి చెప్పండి ఎవరన్నా తెలీదా తెలియట్లేదా
Sir, old people too, I can see.
Yeah, you are, please introduce yourself.
Sir?
Introduce yourself.
Sir, Umarani, sir.
Umana, okay. So 2 old people you have seen. Okay, that is far. What about near?
Near 3, I can see 3, sir.
Yeah, you see three people. One,
yes,
one
guitar.
2 picture, 1 person in the ear.
Yes,
a woman coming out of the thing, and
yes.
then you see one man with the guitar, and you see another woman holding her cap.
Yes.
So likewise, so why I put this slide is the observation is very important. So what you look, what you perceive is different. What is actually is different. You should look in such a way that you are going to get the correct picture of it. Don't go by visual perseverance. Sometimes తెలుగులో చెప్తారు చూసినవన్నీ నిజం కాకపోవచ్చును విన్నదాన్ని నిజం కాకపోవచ్చును అందుకని ఏమంటారు ఒక పాట కూడా ఉంది కదా దాని ప్రకారం మనం చూసినవన్నీ నిజాలు కావు విన్నవన్నీ నిజాలు కావు మనం బాగా ఆలోచించి తీసుకునేవే నిజం సో I just wanted to tell you that clear observation and perseverance is important for you to come out with what is the reality in thing. Yeah, one more. This might be the last one. Can you find the baby? How many people can find the baby? Put the chat box. Put in the chat box how many people find the baby. I gave the answer. You need to look at least 10 minutes without blinking your eyes. కనిపించిందా విజయభాస్కర్ ఉన్నారా కృష్ణమోహన్ గారు ఉన్నట్టున్నారు చిన్నలా కనిపించిందా ఎంతమంది కనిపించింది బేబీ ఎవరికే కనిపించలేదా కొంచెం బ్లింక్ చేయకుండా ఐస్ కాసేపు తదేకంగా చూడండి I got something on the chat box.
Umarani, sir.
Umarani, na? Umarani seem to be active as usual. ఇదే ఇక్కడ బేబీ చూడండి can you see baby now? Okay, if you are not able to see, then again you need to look into and see. Vijayabhaskar మాట్లాడాలి విజయభాస్కర్ గారు ఉన్నారా ఆన్ చేసి వెళ్ళిపోయారా విజయభాస్కర్ కనిపిస్తున్నారు కానీ నాకు ఎందుకన్నా మైక్ మ్యూట్ అయినట్టుంది యా రైట్ I thank you one and all for your patient listening, and I hope I have done justice to the topic given to me, and I have seen that the fundamental and boring aspects are not touched upon, and seen that some what is happening around the particular thing, what is going to be, and if any questions are there, probably you can raise. If at all I know, I will answer, otherwise I will refer and tell you.
Thank you, sir, for excellent session and talk on nanotechnology. Have any questions, please? Do you have any questions, please, my co-faculty members?
Questions maybe two reasons. One is everything they understood or nothing they understood. I hope it is not the two. Second one. No.
But you don't know, sir, the faculty, they're not interacting.
I don't know why.
It's the same situation when you go to the class, when you're asking the students interact, but here the faculty is not interacting.
నాకు ఇంకో డౌట్ వస్తుంది ఏంటంటే ఆన్ చేసేసి వాళ్ళ పనులు చూసుకుంటారేమోనే అది కాదు కదా దానికి కూడా ఎవరు రెస్పాన్స్ ఇవ్వట్లేదు చాలా మంది తెలిసిన వాళ్ళు ఉన్నారు నాకు ఓకే ఇంటరాక్షన్ షుడ్ బి దేర్ ఓకే మీరు అడగండి నేత్ర మీరే ఏదైనా అడగండి క్వశ్చన్ ఏదో ఒక క్వశ్చన్ అడగండి ఏదో కుమారాణి అడుగు ఏదో ఒకటి
Prasanthi is there.
ప్రశాంతి కూడా అడగండి ఎవరు ఏదో ఒకటి అడగండి సో దట్ ఎవరు నెక్స్ట్ ఆ తర్వాత ఎవరైనా ఉందా
ఉమారాణి మేడం అన్మ్యూట్ చేసుకోండి మీకు ఇచ్చాము ఆప్షన్ ఆల్రెడీ
సార్
అన్మ్యూట్ చేసుకోండి మేడం
okay sir, thank you sir. Sir, you have touched all the drug delivery systems, sir, and explained very nicely, sir. So we used to listen to your lecture during our MPharm. Now I have got the chance to listen to your lecture, and very nice to see you, sir. And thank you for mentioning our works in this lecture, sir. Today's lecture.
It is my pride. My success is you. You people, you do the laboratory work, you do the thing, and with that only we get the awards and rewards. Right?
Yes, sir. Yes, sir. And you have given various insights into different drug delivery systems as well. Its commercialization aspects also. That is very important, sir. Nowadays, we are doing research work, but we are not thinking about its commercialization because we are still research only we are doing, and after that we are leaving. So further, we are not able to understand how to move this the next step that you have given very nicely explained, sir. So thank you very much, sir.
Sir, one question from the chat box, sir. Nanogel or emogel, which is more useful for youthful formulations? Which is more useful formulations?
Nanogel or emulgel? Yeah, emulgel and nanogel. The only thing is the difference between these two are the nanogel. Both are gel based only, right? So in nanogel, we see the drug is in the form of the nano. So usually the nanogels are more effective than the emulgels.
Okay. This Umarani madam, where she is working? Okay. Where she is working, sir? Umarani madam.
Umarani is in Uma Chapandi. The Women's College in Hyderabad, Secunderabad.
Sir, unmute. Okay, sir. Sir, I am working in RBVRR Women's College of Pharmacy, sir. Barkatpur, Hyderabad.
Okay.
Affiliated to Usmania University.
Tell your principal's name.
My principal, Professor M. Sumakant.
Sumakant.
I have visited their college as an examiner.
Yes, sir. Yes, sir. You came.
Yeah,
for
it was good.
Viva.
All young people are around and things are doing, and our principal is very strict.
Yes, sir.
Good.
So popularly known as Reddy Women's College.
Yeah, Reddy Women's College, correct. Yeah.
Thank you, sir, Gopinath, sir. And you have given very nice lecture in the morning. I couldn't able to give response, sir. And that yoga and everything you have explained very nicely, which is very much important nowadays because we are all in stress now, sir. So after pandemic, the stress is more. like mentally and like financially also?
No, no, madam. That the base for the major base is that one is that you have to identify the and where the root cause for that.
Yes, sir.
And you are not identifying the root cause. You are going for subsiding that one. Okay. Any questions is there? Otherwise, we'll give the vote of thanks to the Professor Nagaraju sir, Dean School of Padmavati School of Pharmacy and University. Sir, thank you very much, Nagaraj sir.
Thank you for the opportunity given to interact.
And Netravani. Unmute చేసుకోండి అమ్మా. Netravani, unmute yourselves.
Okay, sir.
Okay.
Our sincere, heartfelt gratitude to Professor Nagaraju sir, who has presented the whole gamut of information on emerging trends in novel drug delivery research. Sir really explained in detail about the nanotechnology in drug delivery, that is advantages and about various nanoparticulate drug delivery systems. Sir clearly narrated the case study regarding liposomes. Sir explained the challenges regarding NDD, novel drug delivery research, which is nowadays needed essentially. And Sir's presentation on their
Netravani, unmute చేసుకోవాలండి.
Okay, sir. So apart from the subject study that is about novel drug delivery research, and Sir's presentation on their PhD and PG students' research work is really wonderful and it is very much appreciating. And finally, apart from the research work and apart from the subject study, the importance on observation, hard work, determination is really true, which nowadays is needed for all. So this lecture has given finally how better
Please.
So importance on observation, hard work, determination, it is really true, which nowadays is needed for all. So this lecture finally has given how better we can teach nanotechnology, liposomes for UC and PG students. Thank you, sir, for your kind acceptance of our request to energize the registered participants with sound knowledge on emerging trends in novel drug delivery research. Thanks for giving me this opportunity, sir.
Thank
you,
you,
sir. Thank you, sir.
Thank you. Shall I stop sharing the screen?
Yes, sir.
Right, sir. Right. Yeah. Am I permitted to leave?
Yes, sir.
And before leaving, thanks to Professor Gopinath, Principal, for giving me this opportunity to interact all of you, giving me this platform to talk to you, and where it is not just a research work, something where, we can, whatever it is going to be. And I have seen the program schedule, and he has invited different people who are able to impact on the audience. So probably that will definitely help all the young people, people who are there around. Therefore, it is going to be helpful to you. Thank you once again, Principal Gopinath Garu, for giving me this opportunity. And I wish a successful completion of this program, and future also what is going to take up some things like that. Utilize the pandemic situation where we can all spend 1 or 2 hours with young people like around. Thank you very much for the opportunity given to me, and all the best to all the people who are there. And I hope you enjoyed the sessions. Thank you.
Thank you, sir. Thank you. and my co-faculty members, some of the information for tomorrow program. For tomorrow, the 5th day of the FDP program. And tomorrow also from the eminent speaker from the USA. And you have to sharply join in the session by 9:30. Because of the time variation in the IST and in ESA is different, because and for them it's midnight. We requested them and they're accepting our invitation, and they're delivering for the benefit of the faculty fraternity. My humble request to you people, join in the 9:30 exactly. And tomorrow 2 sessions will be there again from the USA. Have any queries, please? Or otherwise we'll end up the session.